postdoctoral training with Professor William Sessa here at Yale University through 2009. Following that, he started his own laboratory in the Department of Medicine at NYU, and he joined us here in Yale at 2013. Since then, using mouse models, cell biology, and genetics, he and his team worked to elucidate the molecular basis of microRNA functions in regulating lipid metabolism and exploring the potential of microRNAs as therapeutic targets. Please welcome Dr. Hernando. Thank you.
Thank you. Well, thank you very much for the invitation. This is an interesting seminar because it's going to be some research, so I'm going to do some research as well at the end. They asked me to talk about the vascular biology program that many of you probably know even better than me. Okay, we have Larry, we have Jeff here. I just started my postdoc here back in 2005 with Bill Sessa, that was at that time co-director of the program. The director was Jordan Power. And then we moved to this building. This is the Amistad Research Building, and this building hosts the 3 interdepartmental programs: the vascular biology program, the human translational immunology program, and the cell stem cell program. Then the talk today, I'm going to briefly summarize how the background and how the vascular biology program was generated, where we are today, and where are the future plans that we have for that. And then I'm going to put an example. It's going to be a little bit closer to me because I understand better that science of how people within the vascular biology program and also the cardiovascular research center, members of the program also, we collaborate together to study, one of the major cardiovascular diseases, atherosclerosis in this case, and also where are the future areas of expansion and collaboration between all of us. Then the vascular biology program, the original program for vascular disease here at Yale was created by Jordan Power, and there were a number of members still in our faculty, members of this particular program, the molecular cardiobiology program that was created in 1991 and was hosted in the Boyer Center for Molecular Medicine until the year 2000, when Jordan started the vascular biology and transplantation program. Then the initials are the same, but the name it was different. Jordan started this program in 2000. In 2005, Bill Sessa became the co-director, and in 2007, Jordan started a new program in the School of Medicine, that is the Human Translational Immunology program, that is tightly linked to the immunobiology program, and Bill started directing the vascular biology program. Since 2008, our program has been tightly collaborated with the Cardiovascular Research Center that was created by Mike Simons when he became the chief of cardiology, and recruit outstanding people here at Yale, some of them here. And Mike was directing the center, I guess, until 2013, where it was led by Jeff Bender. Currently is directed by 5 co-directors, including Catherine Martin, Stefania Nicola Nickman, and Dan Grave. I put these bars together because we actually collaborate quite often. We have shared a seminar series. Many of the labs that are in the vascular biology program also collaborate with the Cardiovascular Center. What is unique about the vascular biology and therapeutics program was how it was originally funded and what is the philosophy of the program, which was to engage basic scientists and clinician physician scientists together in the same building, the same environment, and all this engaging in multidisciplinary groups. At that time, the idea from Jordan was to engage young physician scientists within a group, of an established group, to allow them to develop their careers, their programs. And coming from that came out some of the names that you see there that today are very well established, multi-R1 funded and highly respected investigators, including George Stolidis, Julie Goodwin, and Alan Dardick are an example of that. As you can see, we have a very important representation of surgery, pulmonary surgery, cardiovascular surgery. We have 5 faculty right now in our program. Then this is the philosophy that was brought by Jordan, and is still the philosophy of the vascular biology program. that is engage clinical scientists, physician scientists with the basic scientists within the same place. Then this is where we are right now. This is the largest interdepartmental program for, at Yale University. And this is our, this cartoon shows you the labs that are located in the Amistad Research Building. In the top part, you have the basic research laboratories. And in the bottom part, you have the clinical research leading by physician scientists. Then, since the last couple of years, we have been doing some changes here in the program. Unfortunately, 3 investigators left the program. One, Bill Cesar, who became the chief scientific officer from Pfizer, and Laura Nicholson also left last year. And Greg Tillen is leaving in a couple of months. Then, despite that, we would be able to recruit some outstanding people, including Andres Hidalgo, who is currently here most of the time. Jenny Thu is also a new faculty member. We just welcome to Patrick Davis, who is working in heart transplant as a new physician scientist PI in our program. In blue are the new recruitments. One of them, Stefania Libreros, she's coming from Harvard Medical School, trained with Charlie Serhan, working in pro-resolving lipid mediators, and she's starting the laboratory in July. And we are very close to sign the Lauren Bewer, who is coming from Tufts University, is an extraordinary vascular physiologist, and she works in the cardiovascular health in women, which is something that we want to strengthen in our program as well. Also in the bottom, with the loss of Laura Nicholson, we were able to also retain one, and this is something new in our program. Sam Radedon is just a fellow that graduated the PhD, and the Department of Anesthesiology is providing him, together with Brian Smith, together a unique opportunity to be able to build his own laboratory, skipping the postdoctoral training phase. Then he's mentoring by several faculties at Yale, and also by Themis Kyriakides at the Vascular Biology program. Then we're trying to keep this line of research, and Sam is trying to develop himself. We're going to evaluate this in 3 or 4 years to see where the program goes and whether it's worth it to establish this laboratory in the Vascular Biology program. As I mentioned to you before, the Vascular Biology and Therapeutics program has 58 faculty members. A large part came from the Division of Cardiology, as I mentioned, but we do have also many other laboratories that are affiliated, and PIs, and some of them from the biomedical engineer, who has also a very big presence in our program as well. Then, since I became the director, we organized the program, established a new steering committee. As you notice there is also a lot of presence in this committee from members from the Cardiovascular Research Center here. We do have also a recruitment committee, shown here in the bottom, that basically they search for new faculty members, independent of myself. Then I give my input at the end. They work independent to me. The program is, we can say I have basically 2 co-directors, and I want to emphasize this. I really get the opinion in monthly basis from George Stelidis and Themis Kyriakides. The reason for that is George is a physician scientist, as I mentioned to you, and there are many issues related to that I need to be educated, and he'll always give me input on that. We have the office close together. We are discussing all these issues all the time, and also by Themis Kyriakides, which is also a member and is leading the basic science program there. We do have also mentoring committee. Again, people we are clinical and basic. As I mentioned to you, these different issues are affecting the faculties. Then this cartoon shows you the resources that we do have here, how we are trying to expand this in these blue boxes that we need to is things that we are working on with the new recruitments, and we can provide also to all the cardiovascular community here. For the endothelial cell biology and vascular physiology, you know that the Vascular Biology and Therapeutics program has a endothelial cell core that we're able to provide to the scientific community here several primary endothelial cells, particularly Hubers, HETMEC. We like to expand this to provide hepatocytes and liver sinusoidal endothelial cells as well, working together with the liver center. The recruitment from Lauren Bewer is a target recruitment as a vascular biology program. One of the things that we are missing, particularly when Bill is leaving, is to have a very strong core for vascular physiology, and this is going to be leading by Lauren Bewer. She's an expert in vascular physiology, trained with Brian Isaacson in Virginia, and she will set up all the equipment to do wire and pressure myography, and also to study blood pressure by using radiotelemetry. Then this has been working. Andres Hidalgo, many of you know, he's an expert also in cardiovascular imaging. He's in the process to buy a high-speed intravital system, and licensed microscope system as well, that will be located in the Amistad Research Building. For the metabolic and atherosclerosis studies, my laboratory and also all the laboratories there, we do have large equipment and resources to do all kinds of studies related to lipid and glucose metabolism, study lipoprotein fractionation, use models of vascular disease, and perform lipidomic analysis. We like to establish also a good metabolomic center there. My laboratory recently acquired a GCMS that allow the analysis of fatty acids and cholesterol derivatives and intermediates. And with Stefania Libreros, who is coming in the summer, she's planning to get the best equipment to analyze glycerolipids and pro-resolving lipid mediators using an LC-MS-MS. This is the last technology. It will allow to do these large metabolomic studies as well. And regarding human tissues, with the Greg Teagan, we know is leaving. He was working together with the surgery department. We still, I talked with Sanjay Kulkarni, and one of the things that we're going to keep still in the VBT is the ability to perfuse human organs, particularly the liver and kidney, and also the heart now. Patrick Davis is setting up the system, and the system is already set up for perfusing hearts, mouse hearts, and to do these studies in our program. I think this was very interesting. The whole the utilization of the human organs to study not only biology was implemented by Jordan Power and Mark Saltman, where they were looking for therapeutical approaches to target the human tissue using nanoparticles. They did that using the perfusion equipment that we have there, and this is open for all the people that want to use it. George Telidis, I think, has been a close collaborator from many of us, providing key samples, particularly coronary arteries from people that undergo a transplant, and that allow us to do many of the analysis that we do, lipidomics, gene expression, etc. And today he has a collaboration also with Pfizer to perform single cell RNA transcriptomics for the first time using these samples. This is very unique, as you can imagine. You have to have these specific resources of surgeon just getting the heart out and the right time to isolate those cells alive and perform the single cell transcriptomics. Then the research that we are doing is very many fields. This is some of the examples from some of the physician scientists, basic scientists. There's one common topic that is inflammation, and I will go back later to this. But for instance, we have the studies from Julie Goodwin that is looking more into the impact on the renal disease in the cardiovascular system. particularly the role of endothelial corticoreceptor and with signaling in this aspect. The lab from Yajaira Suarez that is working in the cholesterol intermediates and derivatives and how this control macrophage immunometabolic responses, atherosclerosis in cancer as well in angiogenesis, the interaction between the human immune system and vascular system is led by Jordan Pover, the studies of the third matrix interaction and angiogenesis by Temis Kyriakides. And then in the context of vascular disease, more in the clinical side, many of the studies being led by George Stolidis, looking into the basic mechanism that leads to aneurysm, atherosclerosis, and cardiac alveolar vasculopathy. And recently, very nice data, also the group from Arnar Gerson working on mechanism of mitral valve prolapse that also we are meeting together and try to put together programs and grants to study this as well. Then I'm going to put an example of how this collaboration goes and, and how it's important the environment that we have here. I can tell you the environment that we have here at Yale probably is, I don't want to feel arrogant because involve all of us, but it's probably one of the top of the countries. You go everywhere and the people acknowledge that thing. And the reason is because the scientists here has a very broad expertise and they have been unique in many of the fields in the vascular disease and particularly in atherosclerosis is quite relevant. When I organized the Gordon Research Conference of atherosclerosis, I invite 5 speakers from here and I could invite 10. I couldn't invite more, otherwise they fired me from that. But the reason is because the people are doing cutting-edge research and they are leading really the field. I put this example. This is because it's the major cardiovascular disease and account for most of the death for cardiovascular disease in the Western societies. This is we can say immunometabolic disease that is characterized by accumulation of lipids in the arterial wall, and that is precedes to this chronic inflammatory response that leads to the formation of this atherosclerotic plaque. This is a human plaque here, and this is a mouse plaque here in the right. This shows you that there are big differences between the human and mouse plaques. But usually mouse they don't die of atherosclerosis, but human they do. But they develop large plaques. Then from the basic point of view, many of the most fast use the mouse models, but we also try to extrapolate these findings to the things that we found in humans. And this is with the collaboration with George Stolidis and others. As you see here in this right panel, this disease is being characterized by accumulation of cholesterol, particularly these cholesterol clefts that are formed here. These are crystals of cholesterol in the plaque, and this huge necrosis in the lesion that is characterized by this cellular area. There's no cells here. There's only cellular debris. There's a large accumulation of neutral lipids. This is stained by Olreto, and also a large accumulation of inflammatory cells that came there for a reason. People always blame macrophages, but macrophages are good. They came to clean up and repair the lesions. The problem is when the lesion became chronic and promote this exacerbated inflammation in the arterial wall. Then I'm going to start this presentation showing classic cartoons from the review from Joe Whitson and Chris Glass, and they actually the title of this review was published in 2000, almost 20 years ago. Was Atherosclerosis: The Road Ahead. Then they is basically summarize key concepts of this disease, and then I'm going to tell you how the research being done here by many of investigators in the vascular biology program help to understand a little bit better of how the disease develops and progress. Then one other thing at that time, back in 2000, is, and we know, and this is very well established, is that one of the major factors that initiate the formation of the lesion in the arterial wall is the infiltration of the low-density lipoproteins. We do know that high levels of LDL infiltrates in the arterial wall and are being retained here in the intima, where they are modified to oxidate LDL. One of the things that was not solved for many years is how this LDL is transported across the endothelium to be retained in the arterial wall. And we can see here that work from Bill's lab and my lab really identify some key determinants that regulate this transport. For instance, if you ablate expression of an organelle in endothelium called caveoli, you block the transcytosis of LDL. Then despite these mice, for instance, has pulmonary hypertension, dyslipidemia, insulin resistance, they don't develop plaques. Then this organelle is being required. Here is a staining of the human coronary lesion, and these are the caveoli, these small vesicles here, and these are the vesicles that carry these lipids and move these lipids from the apical sides to the basolateral sides, facilitating the transport to arterial wall. In addition to that, Bill's lab identified the TGF receptor ALK1 as a potential receptor located in the caveoli that bring these lipids in the arterial wall. This is in the context of lipids, but the endothelium we know is highly active and is activated by many factors, including the flow dependent factors, and also metabolic factors, high levels of glucose, dyslipidemia, etc. And here has been a very nice work from Martin Schwartz and Humphrey's lab, identify key determinants that regulate the mechanotransduction in the endothelium, and in this case, the discovery of PKAN1 in integrins that are important, also fibronectin that are important in how the endothelium sends the flow and how the endothelium transmit this to activate inflammation, the inflammatory response. And this is important because when we have these lipids, and we know that these lipids in a classical view are the ones that activate the endothelium, right? Activate the endothelium to promote the expression of adhesion molecules that leads the infiltration of inflammatory cells to repair the lesion. We know that it's not only the lipids that do that thing, but also other factors and very important determinants are the flow, for instance. And in this aspect, Martin was able to contribute very importantly to this particular field. Back in the early, maybe in 2013, 15, more or less, it came out this concept that is the endothelial to mesenchymal transition in response to oscillatory flow and inflammatory cytokines. This was very interesting, very important finding by Mike Simon's laboratory, where they found that when you stimulate the cells, endothelial cells with an oscillatory flow, or even with inflammatory cytokines, they change the fate. Then the phenotype change, they start to express mesenchymal markers. They lost endothelial markers like enos going down, and also these facilitate a more favor a more pro-inflammatory phenotype. This was not only shown in mouse, has been shown also in human plaques. Then the endothelial cell fate changed in response to these pro-arterial factors. Similar results were found by Jason Kovacic and other groups in Mount Sinai, and this appears to be very important. And the last factor I want to point out here, probably there are many others, and this is now a hot topic in research, is what's happening in the endothelial metabolism. This is very interesting because we thought that endothelium is somehow quiescent and they are not being affected by many metabolic groups, but we do know now that, and I will touch this later on, that the metabolism itself is important for the inflammatory response upon incubation of the cells with pro-inflammatory cytokines. And you probably know the work from Yale Scalzo and others that if you stimulate cells with TNF, I1 beta, they became glycolytic, and the utilization of glucose is important for this pro-inflammatory phenotype in the cells. And looks that when you use fat, in this case, stimulate fatty acid oxidation, you have the opposite phenotype. Then it's now a very interesting time in the field, looking that the endothelial metabolism is not only important to maintain the function, but also the fate and phenotype and the activation appears to be fundamental, the regulation of these metabolic pathways. And in this case, there are a lot of work going on right now here, including for the groups from Simons, Yajaira Suarez, and myself as well. Then we learned a lot about the endothelium. And then I move on in the process of atherosclerosis. And when we have these lipids here, activate, but not only activate the lipids, other factors activate the endothelium, promote the infiltration of the monocytes. And these monocytes differentiate to macrophages. And these cells are very interesting and very unique because when they differentiate, they express these receptors here that are called scavenger receptors. And very interesting receptors because the expression of these receptors facilitate the uptake of all these lipids. And these receptors are not being regulated by cholesterol itself. Then this is not go to the theory of Gaussian and Brown. These cells can take cholesterol in a massive way, and they can take this cholesterol and esterify this cholesterol and accumulate in these large droplets. This is made by the enzyme called ACAT, and they accumulate these lipids. And then when they accumulate these lipids, these cells are trapped this in the plaque, and the massive accumulation of lipids can eventually lead to ER stress and lead to the death of the macrophages in the deposit of these lipids. Then one of the things that we have been working on very actively here is how we can remove these lipids from these foamy macrophages. And to do that, the macrophage express a number of receptors, and particularly important is one that's called ABCA1 that pump cholesterol and phospholipid out to these other lipoparticles called SDL that we do now play a protective role in cardiovascular disease. Then this is important, and we have been contributed here to elucidate key mechanisms that control the cholesterol efflux pathway, particularly the role of microRNAs and other factors as well. And another thing is how these macrophages not only are important in scavenging these lipids, but also to modulate the inflammatory response in the arterial wall. And in this case, the work from Jeff Bender looking into RNA binding proteins and how this regulated pro-inflammatory cytokine production has been fundamental for many years, as well as now the contribution of some of these key lipids that accumulate in these cells that actually direct the inflammatory response of this macrophage in the plaque, particularly some of the key lipids that you can find in the plaque, desmosterol, lanosterol, and the 25-hydroxycholesterol. Then we are advancing in the disease, and now we have more advanced disease here, where we have accumulation of all these macrophages, and when these macrophages they are largely accumulated in the plaque, also these macrophages control T-cell responses, and this is important. And I mentioned before Jeff, but many of the cytokines that are being produced by the macrophages also are important in promoting the expansion of the smooth muscle cells. And this is key to the plaque formation. This plaque is being covered by the smooth muscle cell layer that we know from L-Vendit many years ago that this should be a clonal expansion of these cells. And one thing that became very interesting in the last few years, and I think very interesting work here from Dan Crave, is where is the fate of these cells, of how these cells are expanded. And the thing that Dan did here, I represent here two very influential papers from him. One of them just published like maybe 3 weeks ago and was the cover from Nature Aging, is using these rainbow mice that allow you to trace the clonal expansion of the cells. Some of the key results that he previously shown are here is that in early stage plaques, you see a monoclonal expansion. That is the thing that we know for many years. You have the media here with multiple colors. You see red, you see yellow, and you see blue. But when the plaque expands and this produces smooth muscle cell migration, you see only a single color, just suggesting that there's only one clonal cell that expands. If you do this analysis in late stage plaque, you can see that you have now many clones. Then there should be some mechanism that trigger not only expansion one clone, but many others. Then I talk with Dan all the time. I try to be a little bit picky with him, and I collaborate with him on this. Because this is beautiful, right? But we still don't understand, which actually means that we have one clone or two clones, three clones, whether this is beneficial or not beneficial for having a more stable plaque or more dangerous plaque. In addition to these studies of clonality, also we have the contribution of Pathy, Martin, and also Arya Mani here that are also looking into epigenetic regulators that regulate the smooth muscle cell plasticity and also the Wnt signaling pathway. And this actually was based also in early studies from Arya linking the alteration of some of the gene associated with cardiovascular disease. Then again, it's not only mouse work, but it's also fundamental work based in the human genetics. There's another aspect here I didn't have time to illustrate here, but these cells don't have only affected by this clonal expansion, but also this overloading of lipids make a change in the phenotype. We talk about the endothelium, how the endothelium change the phenotype in response to metabolism, in response to pro-atherogenic stimuli, but also the smooth muscle cell. We do know that if you load these cells or these cells engulf lipids, they also change the fate. They lost this contractile phenotype, they became more quiescent. And this is also an active program of research here, looking into what is the specific contribution of these metabolic changes that induce these phenotypic changes in the smooth muscle cell in the context of plaque progression. This not only happened in the context of atherosclerosis, but also in vascular aneurysms. This is a beautiful paper by Mike Simons and George Stelides, showing that these cells are highly plastic. You can see here the reprogramming of the vascular smooth muscle cell and how this smooth muscle cell start to spread many different markers that are associated with different cellular lineage, right? We have here a bone crosser marker, that you have here also sulfonin marker, and then you have markers that are associated with bone, adipocyte, and all kind of markers, and suggesting that there is a highly plasticity of these cells, not only in plaques, but also in other vascular diseases. And what actually this means in the context of the progression of the disease is still unknown. And I think in the next 5 to 10 years, we will learn more about this, particularly with the new technology coming out. I think that one thing that will be very interesting is to apply the spatial transcriptomics, not only in vascular aneurysms, but also in plaques. It has not been shown there yet. We're trying to set up here at Yale this technology to see what's actually telling us all this change in the fate of this vascular cell during the progression of these vascular diseases. Then this is just, we can say, latest stage of the plaque, when this plaque broke, and then we had this platelet aggregation and the formation of the thrombi that eventually can occlude the vessel and leads to myocardial infarction and stroke. And here we do have also some outstanding investigators working in this particular setting. I highlight here, John. Well, not because you are here, I didn't know that you are here, but I prepared this before. You didn't call me yesterday, I can tell you. But John has been done, just terrific work here, and not only how platelet biology works in different cardiometabolic settings, including diabetes and dyslipidemia as well. You can find many of these outstanding papers publishing from his laboratory recently. Then when we have this lesion, and it's a very advanced lesion, one I think that we are looking into is how we can resolve this inflammation. And I think here we can have one of the new recruitments, Stefania Libreros, who is working in pro-resolving lipid mediators, particularly how we can resolve the inflammation here. We were close to Thor from Northwestern that is also working in this particular aspect. It's not done yet. We will try again, but it's also was a target recruitment just to look how we can reverse the disease, and particularly how we can attenuate inflammation in the plaque and also in the heart. Then things that we are moving on, and I think cardiology probably is stepping on here just by recruiting Jennifer Kwan, is the relevance of the chip or the clonal hematopoiesis with intermediate potential. I'm going to spend the last few minutes with that. This was the renal paper published by Ben Eber a number of years ago in New England Journal of Medicine that was spun out by many laboratories, and they proved experimentally using different animal models where they found specific mutations in the bone marrow progenitors that predispose them clonal advantage to be populated there. Then you have a clonal selection based in mutations that don't affect you, don't predispose you to have leukemia, but they don't cause leukemia per se, but facilitate the selection of specific clones in the bone marrow. The analysis from Ben Eber here was that most of these genes that affect the clonal hematopoiesis intermediate potential are epigenetic regulators. And here is step 2, and Kathy was one of the co-authors of the renal papers here. The interesting thing of this study came out in one of the supplemental figures, 14, 15, or 16 supplemental figure of this paper, where he found that this clonal hematopoiesis intermediate potential actually is clearly linked to cardiovascular disease and stroke more than anything else, and it's shown here. Several groups, including Ken Walsh, I think Ben Eber as well, they proved the concept in mouse that doing a bone marrow transplantation from mice lacking TEP2, and now they're doing with DNT3. If you do transplantation of bone marrow to dyslipidemic and atherogenic mice, these mice develop very large plaques. And one of the mechanisms that is leading to the formation of this lesion is the pro-inflammatory phenotype of these macrophages that produce significantly higher levels of interleukin-1 beta. Indeed, when they use the antibody that was used in the CANTOS trial, they are able to prevent a little bit the progression of the disease, at least in mouse models. There were many things now looking into that because it became a very interesting thing. And indeed, we were trying to recruit in the BBT also another expert working in bone marrow, Daniel Lucas, looking into the biology of the hematopoiesis. This is actually very interesting, and I put here in the right side things that are not solved. This is the concept that this was associated with aging, that the clonal hematopoiesis of intermediate potential predisposed to atherosclerosis. Is this not entirely clear? Because we do know also that any cardiometabolic disease like established atherosclerosis or even high levels of circulating glucose also promote increase in clonal expansion as well. Then we don't know whether this produce atherosclerosis, or if you have a lesion or tissue damage in the body that talk to the bone marrow and now facilitate the expansion of the bone marrow cells. This increase in the bone marrow renewal also promote the clonal expansion. Then this is not clear yet whether the chip predisposed to cardiovascular disease, or the cardiovascular disease predisposed the chip and this feedback forward to promote or aggravate the disease. And I'm telling you because this is very important. We don't know now, for instance, factors like hypertension, high triglycerides, glucose, high BMI, all these is accelerate the hematopoiesis. And many of these factors that needs to be studied also, including infection. I don't have the time to show here. You infect the mice too, they do the same, actually. And these predisposed changes in the bone marrow that cause or major drivers of the disease. It has been a crazy journey here for many labs, and this has not been only associated with atherosclerosis, but every time that you just check papers in the literature, there have been also clear associations of chip with heart failure, for instance. There are some studies from Fuster and others. This is one of them, but there are others that associate clonal hematopoiesis with heart failure. And not only that, kidney fibrosis, whatever you want to look into, makes sense because these macrophages that are derived from these clones are more pro-inflammatory, and then when you have this exacerbation inflammatory response, usually they aggravate the outcome of the disease. Then in the heart failure, this is a study from Jose Fuster in the CENIC with Valentin Fuster, we actually proved that particular concept as well. And this was done also in people looking for DNT3A, that is the major chip in humans, and TEP2 associated with heart failure. And I point out this because our program right now is really focusing on not only the inflammation in the arteries, but inflammation in the heart, and also the metabolism in the heart. Several recruitments goes towards this direction. One example why the heart is very interesting, is probably the most interesting organ. If you study metabolism, people look into the liver, it's actually the heart, the most interesting organ to study metabolism. And inflammation probably is becoming probably one of the most interesting one. And this is data from Andres Hidalgo in our program that show the relevance of macrophages in the heart. In the top panel, this is the CX3CR1 GFP that track the localization of macrophages that are highly abundant in the heart, as you can see here. This concept is not new. This was known by many years, and the role of macrophages. I'm going to point out where is the discovery of this paper, but probably one of the first papers also published in Cell by Matthias Narendorf, even 3 or 4 years before, was the elucidation of the key contribution of macrophages in the heart in the cardiac conduction system. When you deplete all these macrophages in the heart develop arrhythmias, a wiki-BAP block, and this was due to the loss of macrophages, particularly macrophages in the AV node. Then this was one of the first examples showing that the macrophage is controlling the homeostasis in the tissue. We talked before that these macrophages are important in the context of the vascular damage in the repair of the tissue. But not only that, the macrophages are present in many tissues and are key to maintain homeostatic function in the tissue, and this is particularly important in the heart. Then this is one of the observations that Matthias Narendorf did at that time, but this is another concept that was brought by Andres Hidalgo here and now at Yale, where he came out with this interesting concept that is cardiomyocytes that are full of mitochondria. And this is probably the organ with also all the myocytes that are full of mitochondria that require this to generate energy. The heart has to work 24 hours a day. And then they require a lot of energy production. And then they have out of mitochondria. And the thing that Andres discovered here is that this cardiomyocyte are able to eliminate these mitochondria through these vesicles that they are being secreted by a cardiomyocyte called exosphere. And these exosphere are being taken by macrophages. Why this is in color? Because he used lineage tracing mice that they can track cardiomyocyte in red and they can track macrophage in green. Then you can see here the macrophage that is green, how it's taking up this. And the thing that is very interesting here is that the cargo in this exosphere is mostly mitochondria, dysfunctional mitochondria. Then the key concept here was that these macrophages present in the tissue are very important to clean up these damaged mitochondria. And if you don't do that, the heart inflames and they develop heart failure. There's an important reduction in the ejection fraction. And part of the mechanism is shown there. And this is a number of receptors being required to clean up all these exospheres. Then I'm going now to my place and also Larry, because Larry is one of the key important specialists in understanding metabolism in the heart. And this is important, going back to the lipids and the lipids to the heart again. The endothelium is, when we talk about lipid metabolism, cardiometabolic disease, is the key in many aspects, okay, in many aspects. One of them, and we do know, in the gut, we have the lymphatic circulation. It's important for the absorption of lipids. But how these lipids are processed in the body, the endothelium play a key role here. And this is important because we heard about these bad particles that are generated, right? We had kilomicrons, VLDL, and the generation of LDL that we need to remove in the liver through the LDL receptor. Otherwise, this can accumulate in the arterial wall. Then one of the questions, and probably people still is this not being clarified, what is the role of LDL? Of LDL is we need just to remove it. The thing that we do know is that presence of these particles like VLDL, kilomicron are essential to maintain energy homeostasis in humans. And the mechanism how this is done is by the action of this lipoprotein lipase, this enzyme that is anchored in the luminal sites of the endothelium through this protein called GPIs-BP1. And then this is important because this enzyme breaks down all the triglycerides that are produced by the liver or in this particle when we eat. When this particle is breaking down the triglycerides, provide fatty acids and glycerol to the tissues. And this is essential. It's essential for maintaining heart function that use these fatty acids for oxidation to the exercise, for instance, to make energy, to maintain the thermogenesis in the body by burning these fatty acids in the brown adipose tissue, and also key also to storage these lipids in the white adipose tissue. Then these fatty acids are resterified with glycerol to form triacylglycerol again, and there are storage there. And in a situation of fasting, when we are not eating, we don't have this energy. These lipids can be hydrolyzed, and the lipolysis provide now the release of free fatty acids that now fit to all the system. This is very important because imbalance of this may affect not only many of the functions, including the heart function. And this is not only in the context of homeostasis, this is also in the context of maintaining physiology. Larry was involved in this particular study with Ruta Menditov, and this actually happened also in models of COVID, right? You can see also in patients, right? And when you are infected by a pathogen, even a bacteria or virus, you have two inflammatory responses to respond against this pathogen. And one of them is the It's called the resistant response, where your inflammatory cells produce inflammatory mediators, try to kill the pathogens, but at the same time, the organism has a tolerance response. Then how we can maintain homeostasis upon infection, even COVID or whatever, right? And this paper by Ruslan suggests one thing that is very interesting is like the cross communication between the CNS and the liver. To promote the output, I show you the VLDL, and this is important because the VLDL and suggests promote the triglyceride flux to the heart to facilitate the fatty acid availability to maintain the function under this particular setting. This has been shown also in patients with COVID that actually leads with hypertriglyceridemia, probably by sustaining the function of the heart in these conditions. One of the things that we are doing, and with this I want to finish, is things that we want to bring here. This is a recent grant that we put, hopefully gets funded, final round from the LEDO, that is to understand this. And I want to put to understand this because one of the things that I see the vascular biology and the paper is to understand metabolism, aging, and inflammation, as I told you later. The endothelium, as I mentioned also at the beginning, is not considered TSN layer. Play a key role by modulating the entry of lipids, and also the manage of those lipids by esterifying and releasing them, and the crosstalk from skeletal muscle, heart, and adipose tissue. This group of investigators here, we put together probably the first network that we do have for the first time drivers that leads to the deletion of genes that manipulate lipid uptake, synthesis, and delivery in the tissues in the microcirculation and also in the large vessels. This is important when we talk about heart failure. Is also microcirculatory problem. And then we do have all the mouse models that allow us to control the catabolism of lipids, the entry of fatty acids in the endothelium, the esterification of fatty acids, accumulation of the lipids in the endothelium, and the delivery of these fatty acids to the tissue. This is very important, and not only showing you that thing, but also how the tissues itself communicate with the endothelium to manage those lipids or this energy availability to these tissues is actually a very interesting area of research. If you remember probably a paper from Peter Carmelier doing single cell RNA transcriptomics of the endothelium in all the tissues. If you do this in the heart, most of the endothelial cells, particularly in the microcirculation, in the heart express fatty acid transporters, and especially to provide those lipids to the heart. If you do in the brain, they usually express glucose transporters, just facilitate glucose. And we are trying to understand here where are the metabolic clues and the molecules that cross-communicate to the endothelium to dictate the fate of the endothelium that permit tissue homeostasis. Then this is the last slide I'm going to tell you. This is very closely engaged now for the specialists that people that we have in the DVT and people that we are recruiting. But when I was actually putting this triangle of things, you can put anyone here. We do know the inflammation is key, and the inflammation is being modulated by metabolism. Aging is a key factor for both. Aging predisposed to metabolic loss of metabolic flexibility and chronic inflammation. We do know the inflammation regulates metabolism. Metabolism itself modulates inflammation. And these three factors are associated with the progression of cardiovascular that are linked and are associated, particularly important obesity, diabetes, or dyslipidemia. And we do have very great experts here working in atherosclerosis and vascular physiology, particularly hypertension as well now. We are looking to expand in two different areas that we consider are important. One of them is a stroke. We do have some great researchers here, including Lawrence Sanson. We need to probably work more together in this aspect. For us, and I think one of the key aspects I think that came out, and it's a vascular disease as well. But I mean, probably we can discuss about this, is the heart failure, which I think became very interesting and very important. Vascular disease, metabolic disease, also with a very important inflammatory component as well. And with this, I want to finish, and thank you very much all of you for being here today.
I think the first comment, a few comments, and then maybe just a question. First of all, thank you for kind of sharing with this very diverse group of investigators and clinicians how you can, how the work you do every day, how the work many of our colleagues do every day has a tremendous potential impact on the patients who care for and family. So it is very key, fully very integrated, even if it's mission investment. One of the other comment I would say is, I hope it's not lost on many of our youngest investigators and our faculty here that I do think Yale is unique in being able to have this really tremendous breadth as well as extensive linkages to the molecular discovery work and population policy. And I think more on that in a moment. We are not very good, I would say, as a self-criticism, of pursuing collaborative grants, meaning strategic-focused research networks, DHA, PPG. We've done well with Ladue, but there's obviously more opportunity. And I hope that this opportunity to bring Carlos here, everyone recognizes the tremendous potential we have to bridge multiple areas from the patient and human biology to the discovery. I think we should do better in that area.
I well completely agree. I mean, I 100% agree with you on that. We have been trying. We put a network for the American Heart, but we didn't get it. But we try our hard. No, but I agree. One of the key recruitments that we are doing now is to emphasize on that. I think it's key to put the PPGs. I think the BBT has been a long history to have some PPG funded for many years. I think Jordan Power did that. They still have one with Mike Simons, but I think the PPGs, I think, have two very interesting things. One of them now is to engage junior faculty. Okay, I think as probably people knows, the NHLBI provide a larger budget if you include a junior scientist. And then one of our thoughts is one of our recruitments to put in one PPE that we are looking into working to the immunometabolism together with Andres Hidalgo, Yajaira Suarez, Stefania Libres, and a third potentially. Then we were thinking on that. There are PPEs cooking, and that has been submitted. I do know Martin Swarth and Mike Simons together with Yajaira, they put a PPE, and I think they're going to resubmit again. But there is, as you suggest, I mean, there is many other opportunities. I think we had to emphasize and do more work on that. I want also to tell, clinical scientists, it's very important to work this together. The university has generated a new program. It's called Points Program. I was funded with one of those together with other folks to work in NAFLD. But this is a very interesting program where the Dean Office provides with $200,000 for 4 groups to generate preliminary data to put up PPG. For PPG, we need this, and we need leaders that want to drive that, okay? I think it's important to tell senior people and also people they have to drive these things. You have to put an overall project and take time, but at the end, it's a very satisfying, thing for the investigator. For instance, this Leduc that you say, I basically put mostly myself. Ira Goldberg gave me 6 bottles of wines the other night. It has been very interesting because you learn. I mean, the bottom line is during the process of writing this grant, you learn, you set up the specifics, and you establish collaborations. And I think in any case, it's a win-win. Despite you get the grant or you don't get the grant, you put people together working on the same topic. And in heart failure should be someone working. I mean, I should be someone working, and we can discuss that thing because we want to emphasize on that,
What maybe my question for sure is, so we're very dependent in terms of human translational biology, one of our areas of focus in a section. The access issue, I would argue that without George's work, a lot of this would not be the problem. What do you know to tell us in your role in leadership across the school and university? How is the school and university supporting and investing in mechanisms to gather tissue and how tissue is? Every time I've suggested that we develop an independent program within our section, they're going to push and say, well, it's going to be done in a larger framework, but I didn't see.
Yeah, one of the goals from the new chair of pathology, Chen Liu, was to create a large biobank in the school. He presented this in the leadership group, I think, in the last meeting. And I think they have a very nice web page that you can access to that. But as I think it's important, you have a large biobank, but I think the clinical data associated with the biobank is absolutely key. You want to get meaningful data. Then I don't know how this has been working out. Then there is a lot of interest to do that. And I think Chen Liu is doing to generate these biobanks. We do want to do something in the BBT as well for arteries. I discussed this with George Telidis. We want to generate like our internal, instead of just sharing samples to everyone, to characterize those samples better and then provide those samples to the people. For heart, well, when you talk about heart, right? This is interesting because in this network I learned a lot of these things, and one thing that I learned is how the people in the pen they are using, and they are using quite well, right? Soltarani is a member of this, and also this network we're collecting also with Dan Rader, that has the PEN genomic database, and this database that they have with all the genomic data also associated with cardiovascular outcome, heart failure, and everything is included in the network, and they also has access to those samples as well. Then this probably is a good model to mimic and do it. It's not only about collecting the samples, but it's a model to collecting that samples and also the electronic, medical data to link to that, and be leading by people that understand the disease. I don't think so. This pathology is everything. But I think this has to engage other people. Then Peter Wan, I think was one cardiovascular pathologist that was recruited by Chen Liu and is a member of the BBT. I can actually ask him to see how these things are being done, because I think we need to standardize those things. And I think it's absolutely key. If we want to translate this again, mouse models are great, but if this doesn't happen in humans, we're not interested. Yeah.
We absolutely have to coordinate at the highest levels with pathology, your program, CVRC, and the dean's office, and surgery, both cardiac and vascular surgery. So I think this really should be a priority, and it's going to take some organization to bring people together to really think about it thoughtfully. So I think Eric really hit this one on the head.
I can try to organize these things so you guys want, yeah.
Kind of a wonderful summary for the program. So our human patient stem cell program can bridge basic science and patient sample very well. So we have all the human blood vessel model, human heart model. So that can be a wonderful bridge between a VBT and a cardiology. So besides the PBG program, do you think about maybe some new translational like T32 program to bring trainees together?
Well, the T32, and Jeff is here. There is a T32 in cardiology, as you know, and one of the they asked me to lead, but I'm quite busy. One of the reasons we want to hire Ed Thor here, and we are going to do also by bringing Stefania Libreros, is to have more representation in pathology. Then the idea is to put a T32 through pathology, more focused in the context on inflammation and cardiovascular disease, more vascular disease. But this is in the pipeline. We're going to do that for sure. Chen has been pushing on that, and we're going to do it. Then I didn't talk about therapeutics. This is another part there. It's because you are here. Don't get offended. But it's when we get all these things, how we can tackle the disease by therapeutics. IPS is there, and we are working also in the adult reprogramming as well.
Yeah, thank you. It's a really wonderful network of individual investigators, and from a trainee perspective, it can be daunting if you're not in that world to integrate and gain access to that space. And so I'm wondering, what's your suggestion for our translationally oriented physician scientists who want to get involved, or as we look to recruit these folks, either in physician scientist training programs or other programs? How should they interact, or what are the mechanisms to engage with you?
Well, the vascular biology program, we work on all kind of physician scientists, and I have a personal view of that. My parents are doctors, both of them, and they're physician scientists, and then we discuss this all the time. I think it's very difficult to understand. It's very important to understand the basic of the disease, knowing the disease in humans. And the vascular biology program is unique for that particular reason. I mean, 50% of the labs are led by physician scientists. Many of them are fed by medical students and PhD MDs that they do rotation and spend time there for 1 year, 2 years. And one of the goals I try to push them is just to interchange more. Then the basic scientist laboratories get more MDs as well. Then the same way that the physician scientist laboratories get more basic scientists. Then it's a constant push for that. And we are doing quite well. We get many MDs. Anyone can reach out the vascular biology program, can discuss with me the interest that they have. I can redirect, people to talk with investigators. I think it's very important for me that the physician scientists get engaged with that. This ecosystem is very unique also because physician scientists usually are quite busy, but when they are supported by this environment, they're able to collaborate with many people in the way that if they have a clinical part, they're collaborating with people in my lab, people in other, and this is the reason why they are extremely successful. Yeah, Jeff.
Yeah, so a couple things. You're being it is very important to give our trainees and potential trainees ideas about what's available and what's going on. And you're sort of the central focal point, and you direct people. And many of us here have been have received your directions to talk to people, and then we put them to Carlos and the group at VBT. So I think it's all about communication and informing people. We have three T32s based in the cardiovascular medicine division. One is in implementation science, so maybe it's a little bit out in the end of the spectrum there. I'm somewhat skeptical that at least the NHLBI is going to give another T32 that's oriented towards cardiovascular biology.
And this is why they want to shift that more towards inflammation.
Yeah, so I think that's a good idea. I think, we can make our T32s as translational as we have people who are interested in doing that kind of work. And I'm certainly committed to some slots that are in the very human translational arena within our T32. I think Ed is as well. One thing very important for the whole environment is to know the new institutes and the new programs that are popping up in the university and school. There's a program, I don't know if it's being called an institute. There's a program in inflammation that's going to have a major footprint in the new building on College Street. And I think Ruslan and Akiko are kind of taking the lead in that. You're probably very involved in that as well, maybe Yahira. That's going to be a powerhouse. And then there's the Neuroscience Institute. There's a new building going up on, I don't know, George Street or Orchard Street. And, the Alexion building is also a center for neuroscience. So there are programs and institutes that are popping up that we need to interact with and
I think key things, the key aspect of inflammation is there. And one of the reasons is we want to push for this pool of money and resources. When they talk about inflammation, well, I think this is a key aspect. I highlight those things here. When we talk about it's interesting, got being pushed by different directions that we should have a cardiovascular institute for research integrating all the people, right? And I think it's very clear in the presentation I saw today that there are many people interacting here, right? And this has to have better articulated, not one director, but many divisions, many things, many divisions working in different specific arena, right? I think this is probably the next step. I think, and I will try to do my best there in coordination with you guys. But, we are facing this in the context of, get these new recruitments. I'm aware that cardiovascular is leading us on other recruitment as well. And I think probably in 1 or 2 years, we will go to the next step to say, well, how we can put this together. Obviously, we will need also large resources, but, we can justify that thing. This is being done for, say, inflammation, or it's done by neuroscience. We do have, and this is the reality, we can say a better base here of research to establish a cardiovascular institute than other in neuroscience. We do have much better than that. But you know, when I want to work on that, I want to work with that specific plan. I mean, people want to see a specific plans. They don't want to see, well, it will be nice to put these things together and the other. And this has to be thought. You know, we talk about clinical science, we talk about biobank, we talk about these things. It will have, it's not so like an institute divide in different things that, you know, can be well articulated and functional. And we have to put all these people together, try to put all these people together. We need the resources to do that. But I will push for that because I think this is the future for us. And recruiting new people, because we all age, this is important to know, then we need the next generation coming in. We have been good, we have to be better, and also to try to put all people together. There's no question about that.
I'm going to ask everyone to grab Carlos and back and just a little bit of a plug. Kathy's here, and we'll hear more about this, but on March 30th and 31st, we're doing a cardiovascular research symposium and have invited 5 really outstanding emerging career faculty, of which we hope to recruit at least 1, if maybe not 2, in partnership. And so more on that. Thanks, Carlos.
Thank you.
Yes.
Well, thank you. The problem with this is that you get people offended for truth because you can't talk about it.
I'm going to try hard to